Hello everyone. Today we are going to see one microbiology case study. This is the case study. A 16-year-old boy which presents to the emergency department with a fever of 104 degree Fahrenheit, and here he has a neck pain and right-sided chest pain. So 3 days prior, he was seen at the urgent care clinic for sore throat and fever. So when we performed the rapid group A Streptococcus, that is a GAS test, was performed and it was negative, and further he was sent home with the diagnosis of viral pharyngitis. So on performing the laboratory testing, and chest x-ray was performed, and it shows multiple nodules in both the right and left lungs, and blood cultures has been ordered, and the anaerobic blood cultures bottles becomes positive. So moreover, gram staining was performed, and the blood culture bottle is subcultured into the anaerobic agar media, and it is placed in the anaerobic chamber for incubation. So anaerobes, which represents the absence of oxygen. So while performing the gram staining from his sample, from his sore throat sample, it was find out as pleomorphic gram negative rods. So these are the gram staining techniques result, which indicates the presence of gram negative rods. So it is red or pink color, so it indicates the gram negative rods. And coming to the anaerobic culture conditions, so the obligate anaerobes require special incubation conditions. So only a small amount of oxygen can be toxic to growth of that microorganism. So for this, an anaerobic glove box incubator can be used for these type of anaerobic cell cultures, and the incubator pumps in anaerobic gas mixture mostly nitrogen with a combination of small amount of carbon dioxide and hydrogen, and it contains the catalyst within the incubator space that mainly absorbs the oxygen. And the smaller laboratories may use airtight boxes or anaerobic jars with catalytic pouches that absorbs oxygen. So this is the anaerobic glove box incubator, which contains the gas, the nitrogen along with the carbon dioxide and hydrogen. So it mainly contains only small amount of oxygen is required, even though it is anaerobic condition. So the 4 different types of agar has been used. One is Brucella blood agar, and another one is phenyl ethyl alcohol, and another one is LKV, that is lacked blood, and the antibiotics which will be used kanamycin and vancomycin, and BBE, it is the Bacteroides bile esculin. So these 4 types of agars has been used. So for blood agar, it is the non-selective media, and in this blood agar most anaerobic bacteria will grow, and polyphenyl ethyl alcohol, it is the selective media only for gram-positive anaerobic bacteria, and LKV, it facilitates the growth of Prevotella and Bacteroid species, and BBE, it is a selective for Bacteroides fragilis group. So when culturing the blood bottle culture after 48 hours, we have noticed some colonies have been grown in that blood agar plate. So the blood which will be used in this agar might be of horse or sheep. Any type of blood has been used for this Brucella blood agar, and no growth was observed and no growth of aerobic subcultures. So it is the failed aero tolerance test. And microbiology lab results. So when we found from the patient's blood culture, there will be a single anaerobic bacteria colony has been identified, and that bacteria is identified as Fusobacterium necrophorum. So it is the common flora of the mouth, so it is acts as natural habitats. It doesn't do any harmful, and it is the opportunistic pathogen. And this bacteria is associated with dental infections, sore throat, and peritonsillar abscesses. And the most severe association is leading to bacteremia and the dissemination of septic emboli. So this causes the Lemierre syndrome. Lemierre syndrome. So this Lemierre syndrome describes any type of throat infections followed by the sepsis. Sepsis, which is the life-threatening condition, often leads to death of the patients, and it is the rare disease. So only the patients were present with sore throat and progressive neck pain is there. And it also most common infections such as viral pharyngitis or strep throat. So strep throat, it obviously causes the group A Streptococcus, that is GAS, and the clinical microbiology laboratory blood culture is identified as Fusobacterium necrophorum. So then the patient was diagnosed with this syndrome after blood culture was identification with Fusobacterium necrophorum, and the patient received some of the intravenous antibiotic solutions in the veins, that is piperacillin, tazobactam, and he was recovered by the usage of this antibiotics.